Hello, everyone, and welcome to today's webinar. My name is Robert Bonomo. I'm a professor of biochemistry, molecular biology, and microbiology and pharmacology at Case Western Reserve University, and I am very thrilled to be your moderator today. This webinar is part of ASBMB's Breakthrough Webinar Series, sponsored by the ASBMB journals. Specifically, today's session is brought to you by the Journal of Biological Chemistry, one of ASBMB's esteemed journals, which has been publishing high-quality science that elucidates the molecular and cellular basis of biological processes for over a century, and many Nobel Prize-winning experiments were first published in this journal. Before we dive in, a quick reminder. You are more than welcome to submit questions at any time using the question and answer feature at the bottom of your screen. We'll address them during the dedicated Q&A session after the presentation. Please keep in mind that this webinar is being recorded, and the recording will be available on the ASBMB website. It is now my pleasure and honor to introduce to you a true friend, colleague, Dr. Alejandro Vila. Alejandro is a professor of biophysics at the University of Rosario, Argentina, and group leader at the Institute of Molecular and Cell Biology of Rosario. Trained as an NMR spectroscopist, his research interests moved to the role of metalloproteins in antimicrobial resistance and their physiology in the bacterial periplasm. His group is a world leader in the study of Metallo-β-lactamases. These enzymes encompass structural and mechanistic studies that have been elegantly elucidated by Alejandro Vila's lab. He has also been involved in inhibitor design and protein evolution. During the last decade, his efforts have been devoted to filling the gap between in vitro studies and the life cycle, evolution, and activity of Metallo-β-lactamases in the bacterial periplasm. Today, Alejandro will be sharing his insights into this groundbreaking research in this field. And in addition, Alejandro has been a dear friend of mine for many years, and it is an honor to be the moderator of this presentation. I must also add, in addition to his very high scientific credentials, he is a consummate expert in red wines from Argentina and from around the world. So we have many questions to ask him, and he will give us lots of great answers and recommendations. Without further ado, Alejandro, please join me, and I look forward to your presentation.
Okay. Thank you very much, Robert, for the kind presentation. I would like to thank ASBMB for giving me the opportunity to present our results, particularly Isabel, who was the one responsible, and also Kalsum for the technical support. I don't know if you can see my screen now. Okay. So, mostly the story I want to tell you today is here on the title, and this reflects the contribution of many talented students and colleagues in my lab and throughout the world. I want to recognize, and I will mention some of them during the talk at the very end of the talk. All of you should be familiar with the threat that we are facing today with the increasing dissemination of antimicrobial resistance. And indeed, one of the latest estimates revealed that 5 million deaths in 2019 were associated to antimicrobial resistance. And this is going to be increasing, and the World Health Organization has predicted that by 2050, this would be the major cause of death in the world. And this got even worse during the pandemics because there has been a huge prescription of antibiotics. The main family of antibiotics are the well-known beta-lactam antibiotics that encompass cephalosporin, penicillins, and carbapenems, and the key of their action resides on the fact they are able to get into the membrane of gram-negative bacteria and interfere with the synthesis of peptidoglycan. This is very well known by all the audiences in all textbooks. And you may also know that the main resistant mechanism developed by bacteria is the expression of enzymes that are called beta-lactamases that can selectively cleave the beta-lactam bond in the periplasm, giving rise to this beta amino acid that is no longer active. Beta-lactamases come in two flavors, serine beta-lactamases and Metallo-vt-lactamases, and this is the ones that we are studying, and we have studied for more than 25 years in our labs. They have completely different folds, as you can appreciate. The serine beta-lactamases can hydrolyze penicillins, cephalosporins, some of them hydrolyze carbapenems, and there are several inhibitors available in the market. The Metallo-vt-lactamases can hydrolyze equally well penicillins, cephalosporins, and carbapenems, and there are no available inhibitors in the market. So this is the main problem about these enzymes. We have no way to stop them. And since they are plasmid encoded, they have been disseminating all over pathogenic and opportunistic bacteria. And going back in history, one of the first reports of a zinc-dependent lactamases was on the mid-60s by E.P. Abraham that described a zinc lactamase from Bacillus cereus, a non-pathogenic bacteria, and at that time was mostly a biochemical curiosity. But if we go 50 years later, several beta-lactamases have been found in plasmid, and one particularly scary is NDM1, the new metallo-beta-lactamases that after being described by the group of Tim Walsh 15 years ago has been disseminated all over the world. So my group has been focusing trying to understand how this protein working on the field on why are they so promiscuous in substrate recognition, their catalytic mechanism, and apply this knowledge to design inhibitors, and also understand the ongoing evolution in the clinics of metallo-beta-lactamases. But I don't intend to go into all of these details, mostly the biochemistry and biophysics of MBL. You can find them in these reviews that we have time to write them during the pandemics. We have a lot of time to write reviews by them and are available for you. But I will summarize all this work in only one slide. Something that we found time ago is that when we remove the zinc from the metal side, these enzymes obviously are no longer active, but they are no longer able to bind the zinc. This means that zinc is essential for substrate binding. And this explains the promiscuity in substrate spectrum of these enzymes, in which we don't have a cavity, but instead we have a broad group flanked by two mobile loops, and there's only the zinc attracting penicillins, cephalosporins, and carbapenems. And these zinc ions are not essential for the stability, as you can see, for the structures of the zinc-loaded and zinc-depleted metal side. And we also reported studying the mechanisms that these enzymes require two zinc ions to be active. So this is the active species to be inhibited. But today I want to talk specifically after this introduction about the evolution of MBLs, and the title of this talk was inspired on a review we published in JBC 3 years ago together with Carolina, Juliana, and Robert. And the driving force of my laboratory in the last decade has been trying to fill the gap between the in vitro molecular studies in proteins and what's going on in the clinic. That's the resistant phenotype, because usually we find most of the groups in the world trying to study antimicrobial resistance are split into clinical microbiologists and protein chemists. Oh, this is a real challenge. To do that, to mind and carry about this gap, we have to take into account that these enzymes are active in the periplasm of gram-negative bacteria, where they are translocated and they hydrolyze the antibiotics. And what happens with mutations in evolution? For this, we need to ask this question: which are the evolutionary traits in these enzymes able to confer resistance? And the point is that this is not so easy, because usually we think that mutations are brought and fixed in enzymes to increase the activity. But mutations have pleiotropic effect, and mutations impact at the same time in the activity, structure, stability, the expression level that at the end of the day, they are going to impact the resistant phenotype, the translocation and falling into the periplasm. They can compromise the bacterial fitness and host. And last but not least, we need the zinc to be bound to this enzyme, and the mutation can also alter the zinc binding affinity. So the challenge is how to put this in context in the periplasm. And mutations can also occur in the signal peptide, not only in the mature enzyme, as Alejandro Gonzalez showed some time ago in the lab, is that in the signal peptide of NDMs, there's a lipoboc with a cysteine that activates the lipidation machinery of the periplasm of bacteria, so that NDM is not soluble as other Metallo-β-lactamases, and is anchored to the inner leaflet of the outer membrane. So the cell localization is also key to understand how they work in the periplasm. And the other concept that I want to highlight is that these enzymes require two zinc ions to be active, not only in the test tube, but we show that this happens also in the periplasm. So talking about the zinc, the question that comes next is how much zinc is available? And this is not an easy question to answer, because zinc is required for pathogenesis and virulence on bacteria. And as Eric Scar showed some time ago in a beautiful paper in Science, in an infectious model in mice, here you can see that at the infection site, there's a massive depletion of metal ions, mostly zinc and manganese. This means that the amount of zinc is variable in the different tissues, in the different organisms with time. And bacteria are facing a zinc deprivation caused by the native immune system. And just summarizing in this cartoon, we have MBLs that are present here in the periplasm, a soluble enzyme or membrane bound, and there's zinc around. And when we have the response of the immune system, calprotectin is the main protein present in the neutrophils that sequesters the metal ion, depleting from zinc in the active sites of these enzymes. So which is the impact of zinc depletion? So first, we measure the resistant phenotype by measuring MICs. These are studies of MICs in E. coli expressing different Metallo-β-lactamases, and we can see when we increase the concentration of the picolinic acid, mimicking the effect of calprotectin, we see that the resistance is going down. But this is kind of obvious. This is what we would expect. But we also saw that different enzymes respond differently to the effects of zinc depletion. But what Alejandro did in the lab, he also measured the protein concentration in the periplasm after addition of DPA. And that was very surprising to see that some enzymes, the protein levels, were going down very, very rapidly, showing that apo-MBL, that is, without zinc, are unstable in the periplasm. And this contrasts very much to what we have seen before in the test tube, in which apo enzymes are stable. And here you can see when we treat E. coli cells expressing different lactamases with calprotectin, when we add the chelating agent, BIM2, IM1 is gone. And when we check on the membrane, where by the spheroplast, when NDS is being found, we have a decrease in the amount of NDM upon zinc deprivation. So this also tells us that apo-NDM is more stable than other soluble metallo-beta-lactamases. So we wonder whether this cellular localization, membrane binding, could be providing an advantage to this enzyme. So we compared two metallo-beta-lactamases, NDM, which is naturally membrane-bound, and BIM, that is soluble. And what we did, we swapped the signal peptides for this enzyme, so we could some chimeric variant in which we could have a soluble NDM and a membrane anchor VIM, and we also mutated the cysteine that is provided the anchor to the lipids, so we had another variable of soluble NDM. And I'm telling this because this worked. And here we can see, upon zinc depletion, this is the degradation of the membrane-bound NDM, and the two soluble versions of NDM degrade very rapidly. And the other way around, this is soluble VIM2, and when we have the membrane anchor variant, we can see that upon zinc deprivation, VIM is very much stabilized. So membrane anchoring protects NDM from degradation and zinc limiting conditions. And these are, we regard that these are the conditions that the bacteria are facing during infection when they are challenged at the host pathogen interface by the native immune system response. So we are getting closer to understand what's going on in the real world. But since we are keen on studying the biochemistry, we wanting to understand the structural basis of this degradation. So we had here two proteins, two metallo-beta-lactamases, the soluble NDM that is degrading very rapidly compared to another lactamase that is stable in the periplasm. And we did a classical biochemical experiment in challenging this enzyme with protein escape. And here we can see both enzymes in the hollow form with the zinc. They are not degraded, but in the apo form without zinc, BC2 in the test tube is stable, similar with what happens in the periplasm, but apo-NDM is rapidly degraded. And protein escape is an indicator of the presence of protein flexibility. And this is something that we cannot see if we do crystal structure. But here, if we compare the crystal structures of apo and zinc version of these two lactamases, they look pretty much alike. At that time, I remember I was trained, as Robert said, as an NMR spectroscopy. And we use NMR spectroscopy on the purified enzyme because using NMR, we can interrogate the dynamics residue by residue of this enzyme. This cartoon shows zinc-loaded NDM with these two alpha helices at the C-terminus, but when we remove the metal ion, we see that in apo-NDM, there's a lot of flexibility exactly at this time. This does not mean that the whole protein is disordered or unfolded. There's some kind of equilibrium between the folded form with alpha helices and this disorder here. And clearly this disorder makes this protein susceptible to proteolysis. And this structural flexibility tags apo-NDM for degradation in the periplasm. This also means that zinc binding stabilizes this enzyme by quenching this dynamic. So there's a direct link on the availability of zinc and the stability of these enzymes in the periplasm that these studies reveal. But we wanted to know what happened in the periplasm. This degradation was inhibited by PMSF, that is a very well-known inhibitor of serine proteases. So Lisandro knocked down one at a time all the periplasmic serine proteases. And making a long story short, here is a summary on one of the many Western blot that he did. Here we can see the amount of protein while it's expressed. This is the wild type upon depredation. If we knock down DEC-P, there is no change in the protein level, but if we knock down PRC, now there is a recovery on the amount of the enzyme. So these tell us that PRC is the main periplasmic protease of membrane-bound NDM. But if we have the double mutant of PRC and DEC-P, then we recover mostly all the amount of NDM before degradation. So DECP plays a secondary role in the periplasmic degradation. But who are these two proteases? PRC is a C-terminal protease involved in cell wall remodeling, and DECP is a protease, is a housekeeping protease that is involved in quality control of misfolding and aggregating protein. But now the focus is going to be on PRC. What we also studied by NMR is the interaction between a mutant of PRC that is inactive but is able to bind the substrates, and we could see that APO-NDM was binding to the active side of PRC. And this was not the case when it was metal-loaded. So this means that clearly PRC is able to recognize this mobile fragment at the C-terminus that is elicited these dynamics upon deprivation of the zinc ion, giving rise to the degradation in the periplasm. I know that's a lot of information, so I will be summarizing these. So what we can tell about the physiology and the life cycle on these enzymes in the periplasm. When we are in zinc-replete, zinc-abundant conditions, usually we have these proteins that are synthesized, translocated, bound to the membrane, loaded with zinc, providing resistance. But when zinc is limited by the action of calprotectin, now we have an accumulation of APO-NDM that is recognized and hydrolyzed by PRC. On the other side, DECP, I'm not showing here the results, is taking care of some misfolding, non-metallated aggregates on the periplasm. So we can have here a whole cartoon showing of what's going on when bacterial cells during the infection are challenged by zinc deprivation. What about the natural evolution of NDM? Because if we look on what's going on in the clinics, there's a lot of variants that are being accumulated by mutations. And here I'm showing IMP-RBIM have almost 100 variants that have accumulated that shape their catalytic efficiency. In the case of NDM, now there are 78 variants. That's a lot because this is the newest among these Metallo-β-lactamases that has been identified in the clinics. But what are these mutations doing? Let's go back to the clinic and trying to understand why these mutations are being fixed. Because if we are trying to synthesize inhibitors, we should be able to target all the variants that are being fixed in the clinics. And this is the evolution. This is not complete along the years of the different NDM allelic variants. And that's a lot of people from my lab who have worked trying to understand what's going on. This clearly a lot of work summarized in only one slide. So I want to thank Guille, Julia, Steffi, Francesco, Jasmine, and Clarissa for this. To our surprise, all of the mutation in the natural NDM variants have been accumulated outside the active site. If we test the resistant phenotype or the activity or the purified mutants, they have similar activity. So at first sight, it is not obvious why these mutations have been fixed. But as I told you before, it is very important to interrogate what's going on with the resistant phenotype upon zinc deprivation. And this is what we did. We interrogated, here is the result with E. coli cells expressing NDM1. This is the MIC reflecting the resistant phenotype. And as I showed you before, this is how the resistance is going down when we increase the concentration of the chelator and we reduce the available zinc. If we compare this with other cells expressing another group of NDM variants, we can see that most of the NDM variants are more able to resist zinc deprivation than NDM1. And this was really a big, big surprise. And most of the mutants that we identified have this methionine to leucine substitution outside the active site that enhances the zinc binding affinity by 2 orders of magnitude. This means that in the periplasm, we have the disequilibrium between zinc-loaded and zinc-unloaded enzyme is being displaced, protecting this enzyme in the periplasm toward degradation. But there are other mutations that we have observed. Here, what we are seeing is no DMIC, the resistant phenotype, but the protein levels. This in red is NDM1, and we can also see that some mutants are degrading much more slowly. And here we can see the average half-life times. I won't be going into the details of all the mutants that we have studied. I will also show two particular mutations. One mutation, that's the alanine to valine mutation that is present in NDM6 and NDM15, is located on the alpha helixes that I showed you before that were prone to be more flexible upon zinc removal. So we did the obvious experiment. We studied this mutant by NMR. And what we have seen is this is the wild-type mutant without zinc, in which the C-terminal is completely disordered and prone to degradation. But in this mutant, alanine to valine in the C-terminal, even without zinc, the C-terminus is stable and the alpha helices are stable. This means that this mutation plays the same role as zinc binding, quenches the C-term dynamics, stabilizing the enzyme. And we had another mutant, a glutamate to lysine, that is present closer to the active site, but remote to the alpha helixes, but is playing exactly the same role because, again, doing NMR, this is wild-type ApoNDM1, and this is ApoNDM9, including this mutation in which, again, the dynamic is being quenched, and this is a distant effect that is being transmitting along 15 Armstrong in the structure. That's an increased stability. And here, when we are talking about stability, it's not the classical thermodynamic stability that we measure on purified enzyme, but it's the kinetic stability in the periplasm that tell us about the half-life in the periplasm of this enzyme. So, one important conclusion here is that the allelic variants of NDM can gain fitness and confer resistance, but optimizing the performance upon zinc starvation, optimizing the zinc affinity, or optimizing the stability against the proteases and escaping degradation. And that's a completely new paradigm in the evolution of metallo-β-lactamases. But trying again to put together these two worlds, what's going on in microbiology studies and NMR studies, the next challenge that Lisandro asked to me is, can we study protein degradation in the periplasm by NMR? And that's really challenging because we have to deal with living cells. And usually this is the NHSQC when we are doing regular NMR in vitro in purified protein. For those who are not familiar, each cross-peak tells us about each amino acid and what's going on, the structure, the dynamics, providing a structural fingerprint of the protein. But the challenge is, can we do that within the cell? Yes, we can do that. Because what we can do is to label specifically our protein with different isotopes, stable isotopes, and then providing atomic resolution instead of being in the test tube in the whole cell. These isotopes are illuminating where our protein is, and we can interrogate them residue by residue. And this has been pursued in the last decade by different groups, but we have, to our luck, we have Andres Binolfi, who is a colleague in Rosario, who's an expert in in-cell NMR, and he, together with Lisandro and Francisco, a postdoc, help us to work to study what was going on in the periplasm, and which is the additional challenge of doing in-cell NMR in the periplasm to study this process. First, this periplasmic space is very small. The protein concentrations are low in the periplasm, but we have good news. NDM1 is membrane bound, so we won't be able to see NDM by NMR because it's rotating slowly. But the peptide fragments are small and soluble, so we may be able to detect these fragments while not seeing the protein, and this means that the spectra is going to be cleaner. But we have the challenge of having one substrate and two proteases, but Alejandro sorted this out by designing these clever experiments. We have the expression of NDM in normal medium, and we induce the expression of this enzyme, and we are recording an overexpression of NDM provides this is crap, honestly. These are all metabolized from the proteins, but this is because we were not having good level of proteases. If we could separately express the two proteases from NDM, hops, here we got, please believe me, the non-NMR experts, that these are new C-termina coming in from the protein. So we could detect degradation in the cell. These were great results, but this was only the start of a project that took us from 3 to 4 years. What we are identifying here is when we have the cleavage of the proteases, the amide groups are turning into a carboxylate, and these carboxylates are located here in this region that's indicated in cyan. But the challenge now we can see them, is to assign this signal. This means putting this label to mean which residue they correspond. And we did that by different experiments that are target, doing selective labeling, and what we finished ending up is identifying the residue to which these new C-termini of the peptide fragments correspond. And here we can see that half of them correspond to alanine residue, some valine, isoleucine, and threonines. And we were very, very happy not only to identify the C-termini, but also to assign like 80% of the different peptide fragments that were released in the periplasm. That's very, very, very challenging. So, but here what we are having in the next, in this cell, as I told you before, is PRC and DECP working at the same time. Even if PRC is the main proteases, so we work in the knockout mutants to see which were the outcome of PRC and DECP acting independently. And here we can see in knockout DECP cells that the cleavage pattern of PRC is much more simpler, leaving only alanine, and valine is much more selective, while DECP, here we can see in E. coli cells in which we knock down PRC, is less selective, so they have different specificities. The second, you might say, the second thing is, okay, you've done a lot of work. You could have done this in the test tube with purified proteins. And obviously we did that. And if we repeat the same experiment with purified NDM and exposing them to the separate proteases, to make a long story short, here you can see that they have a different pattern in vitro than in vivo. So if we're going and interrogating what's going on in the periplasm. And we could also confirm at atomic level by NMR that PRC is the predominant proteases in the periplasm, as we have observed before in the knockout experiments. But most importantly, we could recognize that DECP is cleaving the peptides that are released by PRC, being a secondary protease. What about the specificity? Because I told you that mostly PRC is recognizing and cleaving alanine and valine residues at 20 sites, but there are more than 20 alanine and valine residues in the sequence of NDM. And most cleavage sites are located in loops and beta strands. So these tell us that in the periplasm, PRC recognize specifically not only the residues, but these residues in specific secondary structures. And going back, to the C-terminal, in which originally in the zinc-loaded protein we have alpha helices. These alpha helices are destabilized upon metal removal, and this is how degradation starts on specific alanine. So summarizing, this is the cartoon that summarizes the whole model at atomic resolution in the periplasm upon zinc deprivation. We can see that NDM is being destabilized and the C-terminal recognized by PRC, and that the products that are released with a very high degree of specificity and they're chopped by DECP, giving rise to smaller peptides in the periplasm. So as a summary, I hope I have been able to convey the main ideas that zinc is essential for metallo-beta-lactamase stability in vivo, in contrast to what happens in the test tube. Protein dynamics regulates the kinetic stabilities of these enzymes in the periplasms, and this kinetic stability is optimized in NDL evolution under zinc starvation. And two general conclusions. Knowledge of the protein physiology within the cell is essential to describe the stability of these proteins, and we have been able to dissect the mechanism of protein quality control by two proteases in the bacterial periplasm in live cells by NMR. And so this is a general challenge for all biochemists. We can and we must fill the gap to get better solutions. So finally, I would like to thank the people who have worked. That's a picture of the group now. I have mentioned those who contributed to this specific work. This is Andres, my colleague, Robert Bonomo, and many co-workers all over the world that help us work. Thank you very much for your attention, and I'm happy to take questions.
Thank you, Professor Vila, for an outstanding lecture. We have some questions in the Q&A part. What I would like to do is that if I could read those questions out to you, and we can decide, the best way to answer them. So let me bring up the first question. One of the questions are, from the point of evolution, is there a disadvantage to evolving more and more stable NDMs? Professor Vila?
Well, there's two answers for that. First, what do we mean by more stable NDM? Usually, if you are measuring the stability by DTM in the test tube, sometimes this type of stability has entailed a trade-off with the dynamics. So that's a problem, because a more rigid protein may not be able to turn over different antibiotics. The second part, if we think about the stability within the periplasm, I think yes, there is an advantage for the cell, because we can have more active protein that at the end of the day is going to provide a stronger phenotype. But we need to study more systems. This is just the tip of the iceberg on one particular protein. That's a very good question.
Okay, I have another question. You presented a very nice story on the NDM, and you showed the importance of the two proteases. But in other species of bacteria, like Acinetobacter or Pseudomonas, there could be a different set of proteases, and there could be different Metallo-β-lactamases. Clearly, your work sets the paradigm for this, but what are your insights or thoughts about studying the various processes in other, let's say, non-fermenters versus fermenters that possess Metallo-β-lactamase? I hope that question is okay.
Oh, that's a fantastic question. I don't know if it comes from you, Robert. So I think that the next step is probably what you have raised, is going to whole specificity, because this set the basis to study what happens in E. coli with one particular set of Metallo-β-lactamases, the NDM. But we have also studied how different organisms are able to host different MBLs, and with different entail fitness costs to the bacteria. And clearly, as you mentioned, DECP and PRC are conserved in Acinetobacter baumannii and Pseudomonas aeruginosa, but non-fermenters have other periplasmic proteases, and they might have different mechanisms. So there might be, here we can think about a co-adaptation. I wouldn't say co-evolution, because that would be too much speculative, but some enzyme could be more fit within the periplasm of some specific bacteria to avoid the recognition of SARP proteases. So that's something this should be the next step, clearly.
Okay, there's something in the Q&A. Let me see if I could, okay, we have a question. Does increasing zinc levels in the blood by taking zinc supplements, for example, enhance resistance? Should we not take zinc supplements during a bacterial infection? And if I recall that I think there's an over-the-counter medicine that has zinc in it that's used for the quote-unquote common cold. But Ale, what are your thoughts about zinc supplementation and etc.?
Well, the issue, there's not an easy answer for that, because when we talk about zinc, I mean, Eric Scar in Vanderbilt is being studied how zinc is distributed among the different tissues, organisms, and that even if we take zinc supplements, this is not guaranteed that the infection side, we're going to have higher zinc, because calprotectin is like 50% of the protein in neutrophils. And the affinity of calprotectin for zinc is very, very high. So what really counts is not the total amount of zinc, but the zinc availability and also the zinc distribution in the different tissues. So it won't hurt, but that's something that we need more experiment, also to do some experiment in vivo with animals to see how this affects, to be able to address this. Also, on the other hand, can we use chelators? That's a very good question, and we don't have a good answer in vivo for that.
Yeah, I think there was a number of years ago, a paper was published in AAC where they used chelators, and it influenced the outcome of a Metallo-β-lactamase bloodstream infection. So maybe it's worthwhile to go back to look at that work. I think it was done in Japan, if I'm not mistaken.
Yeah.
There's another question, Ale. Are you okay with one or two more questions?
I'm happy to take all questions that are available within the allotted time.
Right. One of the participants asked, what other technique could have provided the same information that NMR provided, apart from cryo-EM and X-ray crystallography? So could you have gotten to this using another method, maybe?
Well, if we are talking about in-cell experiment at atomic resolution, we cannot do X-ray. I mean, for the purified proteins, NMR is not the best technique. We go for cryo-EM, for less system, we go for X-ray crystallography, and we do that in the lab. But to interrogate what's going on in the cell at atomic level, NMR is the only technique that can do what I have shown in the last part of my talk, and that's the power of NMR. Okay. And we do that in live cells. That makes the difference. It's not periplasmic extracts.
All right, there's another question regarding, the question was geared towards protein evolution. Was the protein evolution data that you nicely presented obtained through bioinformatics, or was it wet experiment? I think go ahead.
No, the allelic variant that I mentioned, the 78, are all for clinical isolates, are real world evolution that have been fixed by the use of antibiotics. So these tell us that we are studying something, all the variants that happened in the clinic to understand which are the evolutionary pressures. And the surprising thing is that it's not just the type of antibiotic. The conclusion is that in the clinics, enzyme variants are currently evolving to overcome the impact of zinc starvation. So it's based on clinical data.
So I'll see if I could circle back and ask a particular question. So modulating the activities of the proteases would be a way to modify or modulate the activity of NDM, in the periplasm. Has anyone ever thought about using, specific agents that affect protease activity as an adjunct to beta-lactams or other antibiotics to overcome MBLs? Is that something that is a logical thought, or because it seemed like the proteases had a very big role in the steady state expression of NDM, at least?
Yeah. I think that's a good idea because clearly when losing the zinc reveals an Achilles heel of these proteins in the periplasm. But now there are some variants that are able to escape the action of these proteases, like the alanine to valine mutation. Even without the zinc, they are not well recognized by the protein enzymes and degradation is much slower. But I think this is a strategy that could be explored instead of tackling chemically inhibiting these proteins, is tackling the stability in the periplasm. And surely the way to go is to understand their physiology.
All right. Thank you very, very much, Ali. I'd like to take our program to a close. I want to thank you very, very much, Professor Vila, for an outstanding presentation and handling all these questions so expertly. I would like to also thank our audience for their kind attention and support of this series. Please be sure to keep your eye out for upcoming webinars for ASBMB on their breakthrough webinar series. And it's now at the closing time for this presentation. And I want to thank Alejandro very much and thank our audience for their kind attention and participation, and thank ASBMB for support of this excellent series. Everyone have a good day.
Thank you, Robert. Thank all who attended this webinar, and thanks to my coworkers.